So, good morning, everyone. Today I want to tell you about a research project that I conducted in the University of Luleå in the very north of Sweden in collaboration with Stockholm University and the startup in Umeå. So on the picture you can see a very typical Swedish landscape where deciduous hard trees have been almost completely suppressed to let room to conifer plantations. And those plantations are of Pinus sylvestris and Picea abies, and those two species together make up more than 80% of the total Swedish tree cover. And the wood harvested is used for timber, pellets, ethanol, paper, packaging, you name it. Harvesting is done by clear cutting, and regeneration is done by planting seedlings. So there are a lot of seedlings, conifer seedling nurseries in Sweden. And some 20 years ago, two Swedish researchers found that arginine, which is an amino acid, is a good fertilizer for conifer seedlings because it promotes good growth and it results in lower nitrogen losses than fertilizer containing ammonium or nitrate. So as you can see on the picture on the left, arginine has a better uptake rate, and also in nurseries they apply multiple fertilizer application, and each application less nitrogen is recovered. But when using arginine, which is the one in blue on the right side, the recovery is better. So they started to commercialize a fertilizer containing arginine, but there are two main problems. The first one is that arginine is only available at pharmaceutical grades, which make it very expensive. And the second one is that arginine is not sustainably produced. So the objective of this project was to see if we could produce arginine by fermentation using waste from forest-based industry as raw material. And the first thing we need to do that is to have a microorganism. And we decided to use a bacteria called Escherichia coli. And what does a bacteria do? It takes up sugar and converts it to many different products, and one of those is arginine. So we did some genetic modification, trying to improve the part of carbon going to arginine instead of other products, trying to prevent arginine being degraded into other products, and also suppressing the natural mechanism that stops the production once a certain concentration is reached. So we got many different mutants that we tried in bioreactors. So bioreactor is the equipment you see on the picture, and inside we can control the pH, the temperature, and the agitation. So these are some of the mutants, and the last one was much better, producing more arginine at a faster rate and with a better yield. And the first mutant produced 0 grams per liter of arginine, and the last one produced almost 12 grams per liter. So once I had a mutant, I decided to look at the nitrogen supply, because as I said, arginine is a good fertilizer, and the reason for that is that it contains 4 atoms of nitrogen. So if we want the bacteria to produce arginine, we have to give it nitrogen. So again, I used bioreactors, and I screened 8 different nitrogen sources, and 4 of them were better in terms of production and productivity. So I decided to continue with those 4 and try to see if there was an optimal nitrogen concentration. So if we give too little nitrogen to the cells, they don't produce a lot of arginine, which is what you can see on the leftmost picture. If we increase the nitrogen concentration, we can increase the arginine. But if we increase it too much, it becomes toxic for the cells, and the production is not increasing anymore. And even worse, the productivity is dramatically decreasing, as you can see on the right. So finally, I decided to look at the carbon supply, so what kind of food I could give to my bacteria. And wood is made up of cellulose, hemicellulose, lignin, and extractive, and cellulose is a chain of glucose unit, and hemicellulose composition varies depending on what tree species we're looking at, but it contains glucose, galactose, mannose, xylose, and arabinose. So I tried these 5 sugars in mini tiny bioreactors, and I also tried a mixture containing all 5 sugars. And the total sugar concentration was almost always the same. And it was very surprising because the best option was actually to use a mixture of sugar, even though some of the sugar by themselves, like mannose and galactose, did not give very good results. So this was really interesting, and once I knew that, I started to think of what kind of industrial waste stream I could use. And the first one I tried was a waste stream from a company producing viscose. So viscose is a fabric, if you don't know. And this company uses wood chips and converts them into cellulose fiber, which are then transformed into viscose. And when they make this cellulose fiber, they produce a waste that is this white grayish thing that you see, that is called fiber sludge. So we can take this fiber sludge, put it in a bioreactor with enzymes and water, and warm it up and stir it for a couple of days. And it produces this yellow liquid called hydrolysates. And these hydrolysates, in that case, contains a lot of glucose, a little bit of xylose, and some mannose. So I did analysis to know the exact... Yeah, I did some analysis to check the exact concentration of each sugar, and then prepare a synthetic solution containing exactly the same concentration. And I compared the synthetic solution with the real waste. So this is how it looked in the bioreactors. The plain line are the result with hydrolysates, and the dashed line are the control. And in gray, you can see that the cells are growing better when using the control. But what interests us is the blue line, which is arginine. And it was actually better with the hydrolysates, which was really nice. And we don't exactly know why. So then what are the next steps? Well, first of all, we need further strain improvement. And we started with that. So we did some metabolomic studies where we look at everything the cells are producing inside and outside, compare different mutants, and try to see differences to see what we could improve. And we got some ideas that we haven't tried yet. And then we need to further improve the process in terms of pH, temperature, oxygen supply. This is just an example picture. I did that already a little bit. But it's very much mutant dependent, which means that we have to do it again with the last mutant that we will use. And then the plan was to try scaling up the fermentation and the purification in a demo plant and see how it's going. And then also we have to test this arginine in laboratory and in fields to see how it helps seedlings growing. So thank you for your attention.